first combine with antigen and then undergo activation. Once activated, they secrete cytokines that act on B cells and cytotoxic T cells that have also bound antigen. This is very important point. With only a few exceptions, B cells and cytotoxic T cells cannot function adequately unless they are stimulated by cytokines from helper T cells. We shall deal with helper T cells as though they were a homogeneous cell population, but in fact there are several subtypes of helper T cells distinguished by the different batteries of cytokines they secrete when activated. By means of these different cytokines, they help different sets of effector lymphocytes as well as macrophages and natural killer cells. Finally, it should be noted that some of the cytokines secreted by helper T cells also act as inflammatory mediators. Let us discuss lymphocyte receptors. To repeat, the ability of lymphocytes to distinguish one antigen from another is determined by lymphocyte receptors. B cell receptors, as noted earlier, after activation, B cells proliferate and differentiate into plasma cells, which secrete antibodies. The plasma cells derived from a particular B cell can secrete only one particular antibody. Each B cell always displays on its plasma membrane copies of the particular antibody its plasma cell progeny are able to produce. This surface protein, that is glycoprotein, acts as the receptor for the antigen which is specific to it, also called constant region and variable region. The constant segments of the heavy chain forms the base of the antibody molecule. Plasma B cells produce only 5 types of constant segments. These form the basis of classification scheme that identifies antibodies as IgG, IgA, IgD, IgE or IgM, as we will discuss in the next section. The structure of the constant segments of the heavy chains determines the way the antibody is secreted and how it is distributed within the body. For example, antibodies in one class circulate in body fluids, whereas those of another class bind to the membranes of basophils and mast cells. The heavy chain constant segments, which are bound to constant segments of the light chains, also contain binding sites that can activate the complement system. These binding sites are covered when the antibody is secreted, but becomes exposed when the antibody binds to an antigen. The specificity of an antibody molecule depends on the structure of the variable segments of the light and heavy chains. The free tips of the two variable segments form the antigen binding sites of the antibody molecule. These sites can interact with an antigen in the same way that the active site of an enzyme interacts with a substrate molecule. Small differences in the amino acid sequence of the variable segments affect the precise shape of the antigen binding site. These differences account for the differences in specificity among the antibodies produced by different B cells. The distinctions are the result of minor genetic variations that occur during the production, division, and differentiation of B cells. A normal adult body contains about 10 trillion B cells, which can produce an estimated 100 billion types of antibodies, each with a different specificity. Immunoglobulins have a stem called the Fc portion and comprising the lower half of the two heavy chains and two prongs, each containing the antigen binding site, the amino acid sequence that bind antigen. The amino acid sequences of the Fc portion are identical for all immunoglobulins of a single class like IgA, IgD and so on. In contrast, the amino acid sequence of the antigen binding sites vary from immunoglobulin to immunoglobulin in a given class. Thus, each of the 5 classes of antibody contains thousands or millions of unique immunoglobulins which are capable of combining with a particular type of antigen. Now, we will discuss the antigen-antibody complex. When an antibody molecule binds to its corresponding antigen molecule, an antigen-antibody complex is formed. Once the two molecules are in position, hydrogen bonding and other weak chemical forces lock them together. First, let us discuss what is an antigen. It is a substance which binds specifically to the products of immune response, that is antibodies and activated T cells, which are actually produced in response to its entry into the body. Sometimes antigen is also defined as antibody generating material, that is a substance which is capable of generating the production of antibodies. But this definition is actually applicable to immunogen. Region of antigen which binds with antibody is called epitope or antigenic determinant. Let us see the properties of immunogen. For any substance to act as immunogen, it should have following properties. Number 1, its molecular weight should be more than 10,000 Daltons. Number 2, proteins and polysaccharides are better immunogens than lipids. Number 3, it should be unrelated to body. And number 4, heteropolymers are better immunogens than homopolymers. Haptens or the partial immunogens do not cause B cell activation and antibody production. Haptens include short peptide chains, steroids and other lipids and several drugs including the antibiotics such as penicillin. Haptens may become attached to carrier molecules, forming combinations that can function as complete antigens. In some cases, the carrier contributes an antigenic determinant site. The antibodies will attack both the hapten and the carrier molecule. If the carrier molecule is normally present in the tissues, the antibodies may begin attacking and destroying the normal cells. This process is the basis for several drug reactions, including allergies to penicillin. Antibodies do not bind to the entire antigen, but to the specific portions of its exposed surface, the regions which are called antigenic determinant sites. The specificity of the binding depends initially on the three-dimensional fit between the variable segments of the antibody molecule and the corresponding antigenic determinant site. A complete antigen is an antigen with at least two antigenic determinant sites, one for each of the antigen binding sites on an antibody molecule. Exposure to a complete antigen can lead to B cell sensitization and subsequent immune responses. Most environmental antigens have multiple antigenic determinant sites, and the entire microorganism may have thousands. The interaction between an antigen binding site of an immunoglobulin and an antigen is analogous or comparable to lock and